And that was Vicky Vale, reporting live from right outside the super prison Arkham City. Now on to our Science Hour. Today we will be discussing the growth of biotechnology, how it is being used on animals, and the benefits along with the drawbacks of this technology. We will be discussing with our guest Professor Robert Browning from the University of Metropolis. For Dr. Rick Mesh, head of the Research in Biotechnology Department at Elliott College, and Dr. Jack White, a researcher for the Institute of Alternative Medicine. First, Professor Browning, can you explain to us what this technology is and how it works?
Well, of course, Jack. Now, there are two types of biotechnology that are generally associated with animals. We have recombinant DNA and cloning. Recombinant DNA is the process of taking a gene or a desired trait from one organism and putting it into another. The process is relatively simple. And I have a rather crummy model to show you. Budget cuts will do what they would do, won't they, Jack? So here we have a organism that is going to donate one of its genes, and this host is going to take one of that gene, and so it will eventually express this bag's trait. Now, how it works is you begin by isolating the DNA. Right here, you have, this is going to be the DNA of our alien host, and this is the DNA of our bag donor. And as you can see, it has a little gene right here in the middle that we're going to want. And so what you do is you use a little organism called restriction enzymes, and they're like the scissors of the body, really. What they do is they just cut out specific places. And so, in this case, my hands are just going to be the restriction enzyme, and they are going to cut out the gene that we want to put in our host organism. Now, this could be anything to a gene for a growth hormone to make our alien grow faster, because it's pretty small. And now we have our desired gene. And now you use that same enzyme for various reasons that I won't go into, but and then you can just cut our host. This is our little circle of DNA. It's called a plasmid, and so now you have this open space, and you need to fill that make this back into a circle, so you can put it back into our host, our little host organism there. So then you just take it, and through another organism, you can tape that back together. Well, it's not tape, of course, in the body, isn't it? And then you just let it all come together, and you have successfully recombined, hence the name recombinant, recombined some DNA, and this is obviously magnification. And you can put this DNA back inside your host organism, and now this gene will be part of your organism's DNA, and it will copy, and it will be able to, spread, and this organism will be expressing this organism's DNA gene trait. Now, cloning is taking the exact replica of an organism. So what we do here is, first of all, you take a body cell, the cell from any body part of your organism that you want to make a clone of, that you want to make an exact copy of. So here we've got a little body cell from the foot of the organism that we want to make a copy of. And so you'd actually use a syringe, and you'd get just the little cell out, and then you take the cell, and then you'd extract the DNA from that cell. And so let's just say that this is the DNA from our host cell, and then you would extract a cell from a female organism, preferably of the same species. But this is an endangered species here, so we're just going to say that we can't find a female of that species. So you take a female of preferably a related species, and you take an egg cell from it, and you would take out its DNA with a micro syringe, and then its DNA would be gone, and you would replace it with the complete DNA as the body cell, as every body cell of every organism contains the complete genetic code to code for your entire body in one cell. So then you just take the syringe again and put the DNA from you know the organism that you want to be cloned into the egg cell of the organism that is going to give birth to the clone, and then you would put it back into the womb of the host organism, and you would allow it to develop until it is ready to give birth, and then when it does, boop, you've got a clone. Right from here, just bam, now you have two of the exact same genetically identical aliens. So that's how cloning works, and yes.
Thank you, Professor.
Anytime, Jack.
Now, Dr. Mesh and Dr. White, I understand you have varying viewpoints on this technology. What can you share with us about the various ways this technology can be used on animals and what the risks are? Well, yes, of course. In regards to cloning, there's astounding amount of support to be offered to the medical community. For instance, according to James Rubble of the University of Massachusetts in the Scientific American of October 1999, we can clone animals that produce human organs. This is a process called xenotransplantation, and as there is a far less supply of organs than there are donors, this process through genetic engineering will be able to save a multitude of lives. Furthermore, through cloning, it is possible to produce animals with diseases that grow at an increased rate. The Genetic Science Learning Center at the University of Utah says that we can create transgenic animals that are born with human diseases and that grow faster in order to study these diseases so we are better able to fight them in humans. Finally, we can use cloning to increase the rate of farming. This also comes from the Genetic Science Learning Center. By cloning animals that are capable of producing human proteins to grow faster, we can have a much more available supply of products that come from farming. So, all in all, it's pretty clear that through this cloning technology, we can greatly benefit the health of humans. Well, thank you very much, Doctor. What do you have to say on this topic, Dr. White? Well, yes, Dr. Mash here talked about how cloning can benefit the health of humans so much, but we must ask, what about the animals? We have substantial evidence that cloning very rarely possesses any benefit to animals. Nina Mack of the American Anti-Vivisection Society in July 2008 has found that cloned animals have tendencies to have low pregnancy rates. They are more susceptible to disease and often have physical deformities. We have to go into this research, knowing that these experiments are cruel and they have unjust punishment to these cloned animals. Next, the Genetics Science Learning Center at the University of Utah says that cloning has a high failure rate. We spend all this money and end up hurting all these animals for the end result of a failure most of the time. This technology simply isn't worth it. Now, even if you have a success, there are still drawbacks. Rudolf Janisch published in the Genome News Network in 2002 that he found that 4% of genes in white mice that he cloned simply don't work. Now, that may not seem like a large number, but if 4% of genes don't work, that organism is going to have some major problems. This technology is not worth the possible benefits, as remote as they may be. It is harmful, it is unreliable, and it is just wrong. Interesting. Mr. Mesh, what else have we found that using biotechnology in animals is useful for? For one, we can use recombinant DNA to increase food production in animals. For example, Kurt Stelwagen of the University of Guelph published in Livestock Production Science that recombinant DNA can be used to increase milk production in cows. The faster and easier it is to create milk, the more milk we can have. And this is just the beginning of how genetic engineering can be used to increase livestock food production. Aquabounty Corporation has produced salmon that grow twice as fast as normal. We have food growing twice as fast. Means twice as much food, an easy solution to the problem of global hunger. Then there's Lawrence Tilley from the University of Cambridge. He has made chickens that are immune to the flu. Less dead chickens. Through this recombinant DNA technology, we livestock production is improved and mortality rates are reduced. Now, my last point is the point of how genetic engineering can be used to create other useful products. David Cummings of the USDA says that farming can be used to produce human drugs from animals. No longer do we have to face the problem of a not big enough supply of insulin. Our animals can make it for us in their blood and in their milk. Malcolm Fraser Jr. of the University of Notre Dame published in the Proceedings of the National Academy of Sciences that he has engineered silkworms to create spider silk, one of the strongest materials known and a long sought after product. Last but not least, scientists at the Texas Agricultural and Manufacturing University successfully created a clone of a domestic pet. Through this technology, the doors are now open to make copies of cherished animal members of the family. Imagine the potential. Well, it seems there are a wealth of possibilities indeed, Mr. Mesh. Dr. White, any other things you would like to add? Oh, yes, indeed, I do. Those salmon you mentioned, Mr. Mesh, those salmon happen to be a danger. They are a danger to the environment. Scientists at the Proceedings of the National Academy of Sciences estimate that if 60 of these so-called super salmon were released into a population of 60,000 wild salmon, that wild population would be extinct within 40 salmon generations. Those super salmon, they just take what they want. They just take those salmon. They just take the food of those salmon, and that's why those salmon they pose a danger there, to the normal salmon. Yeah, and these salmon also pose a human health hazard. The Ocean Conservancy says that there's an indication that these genetically modified salmon, they have there are indications here that these genetically modified fishes, yeah, they're gonna have a higher level of allergens in these genetically modified salmon than they would in normal salmon. And yeah, so by providing these fish, producing them, sorry, we are heightening the potential for allergic reactions in our consumers. And now, finally, the Cornucopia Institute has found that by consuming genetically modified salmon, we will undoubtedly be consuming the growth hormones that were modified and placed in them. This is a threat that will almost definitely lead to a negative reaction in a human consumer. Now, then, there are the objections that have been put forth by religion. For instance, Ray Epp from the Mennonite Mission Network claims that creation is God's and is not to be tampered with. So, obviously, there are some obligations there. Objections, my apologies. William Bainbridge, PhD, has published surveys in the Journal of Evolution and Technology, and what he says here is he has done these surveys regarding different religions and their views on biotechnology. He found that the majority of Catholics are against cloning, with 74.6% of Catholics discouraging cloning and 59.5% wanting laws against it. Furthermore, he published that 76.1% of Buddhists also discourage cloning, and that 55.7% of Buddhists want laws against it. Very concerning indeed, Dr. White. Well, I'm afraid I have to interrupt now. It appears we have breaking news coming right just outside Arkham City with Vicky Vale. It appears billionaire Bruce Wayne has been kidnapped. Going now to Vicky Vale with the story. This is Jack Ryder from Gotham 9 signing out. See you tomorrow, same time, same channel.